Welcome to our Biomed Unleashed podcast. I'm Alisa, a student specializing in regenerative medicine at Maastricht University.
And I'm Misha, also a student at Maastricht University studying the Maastricht Science program.
Our podcast is endorsed by the Maryland Institute for Technology-Inspired Regenerative Medicine and supervised by Professor Lorenzo Moroni. We aim to make regenerative medicine accessible and understandable to everyone.
In our episodes, we invite scientists, specialists, and innovators to share their career insights and latest discoveries in this exciting field of regenerative medicine.
Today, our guest is Professor Robert Langer, who is an American bioengineer, chemical engineer, and businessman. He specializes in the fields of tissue engineering and drug delivery systems.
Robert Langer is the most cited engineer in history with over 430,000 citations and H-index of 326. Professor Langer co-founded more than 40 biotechnology companies, including Moderna, which is the biotech company specializing in developing messenger RNA vaccines.
Professor Langer has won more than 200 awards, including the Queen Elizabeth Prize, usually referred to as a Nobel for Engineering, and holds more than 1,400 patents. He also founded Langer Lab at MIT, which is the largest biomedical engineering lab in the world with more than 100 researchers. Hello, Professor Langer. Thank you for coming today and being our guest on our podcast.
Oh, it's my pleasure.
Yes, so we would like to start from the first question from your childhood, when you received a gift from your parents. It was a chemistry kit, and you get very interested in chemistry. And then in school, you were good in math, and your parents suggested you to start engineering at college. And you then decided to do a major in chemical engineering. And our first question is, after finishing your chemical engineering degree, how did you get into biomedical engineering?
Yeah, well, as an undergraduate, I majored in chemical engineering. As a graduate student, I also did chemical engineering, but I did a project on enzymes for my thesis. But what really got me, I suppose, into biomedical engineering was my postdoctoral work. I worked with a man named Judah Folkman, who was a surgeon, and working in his lab I was actually the only engineer at Boston Children's Hospital where he worked. And I just got to see so many things. And I suppose that, that really had a big effect on my life and my career.
And later in your career, in the 1980s, you pioneered the field of tissue engineering. Could you explain what tissue engineering is and what inspired you to start developing this field?
Yeah, well, tissue engineering really involves creating new tissues and organs from scratch, which could be, for example, artificial skin for a burn victim. Also would include creating organs and tissues on a chip. So that's kind of how it is. What inspired me was actually when I was at Children's Hospital in the 70s, one of the people I did a lot of work with was a man named Jay Vacanti, and we were good friends. We're still good friends. And he, when he finished his residency and his fellowship at the hospital, he became head of the liver transplant program. And one day I remember he came to see me when he was doing that and said, he's transplanting livers to these little babies and how terrible that was, and there's a liver shortage and they die. And he said, is there any way that he and I could develop tissues and organs from scratch?
And then what exactly he asked you for?
Well, what would happen is, we tried different things. He talked to me about, I had done a lot with polymers and maybe we could create polymers that could do that, which we did, but then we didn't get enough cells in some of the things we did. So we started talking about like branching structures and, so we kept on working together over many years to work out all kinds of different issues, the right materials, the right designs of those materials, and so forth.
So you tried to design something where you could put cells and then implant it in the patient. Okay.
Yes.
Thank you. You have experienced a lot of skepticism and failures and rejections. And in the beginning, no one believed you that large molecules can be slowly delivered to the body via tiny particles. And as you mentioned in other interviews, you found 200 ways that it's not possible to do that.
Sure.
And also your first 9 grants and first patent were rejected. Can you share a specific moment when you realized your work was beginning to gain acceptance and credibility?
Well, yeah, it was sort of evolving. I mean, it took a long time. I mean, I remember finally, after getting all my grants rejected, I got one accepted for about $30,000, and that was very nice. I initially, they also didn't want to. Some of the people didn't want me to stay in the department I was in, when I was a young professor. But eventually they decided to give me what's called promotion and tenure. So those I think helped, getting a grant, getting promoted. So that made me start to think at least I was doing a little better.
And do you think that the luck played a role in all these decisions that other people were making regarding accepting you and etc.?
That luck did?
Yeah, do you think luck played an important role in that?
Well, I think luck always plays a role in everything, but I also think, I remember, I used to play golf when I was much younger. My dad, and I hit a bad shot, and I'd say, I have bad luck. And my father always would tell me, you make your own luck. And I think that's true for everybody to an extent, but certainly luck plays a role. But, I think every individual also plays a role in terms of their determination and so forth.
So you think the personal qualities are the first ones, and then the luck comes and can help this person?
Yes, I think that's well said by you.
Okay.
Okay, and you also often mention that you're stubborn and stick to your ideas when facing failures and rejections. And has stubbornness been a part of your personality since childhood, or did you develop it due to certain influence or experience?
I think if my parents were still alive, they'd probably say I had it when I was a child.
So you think it went through your whole life with you from the beginning?
Yeah, I think so, yes.
Okay. You're running the largest biomedical engineering lab in the world with over 100 researchers and over $10 million in annual grants. Could you please tell us what are you currently working on in your lab?
Well, there are a lot of things with that many people, but to put it into 2 areas, one area is what I'll call drug delivery systems. You mentioned, earlier about the tiny particles that could deliver large molecules. So we're doing a lot on nanoparticles that could deliver, genetic material, nanoparticles that you could target to specific cells. We're working on oral systems that could, you could swallow a pill that could last for a week or a month. You could swallow a pill that might deliver RNA or insulin, which you can't do now. Many different kinds of delivery systems, even helping. We're doing a lot with the Gates Foundation on trying to help, nutrition for the developing world. We're trying to come up with better vaccines, like a vaccine that we call self-boosting, that you'd only have to take once. That's one set of areas. And the other, I would say, would be tissue engineering, organs on a chip, like we're working on a brain on a chip, a GI tract on a chip. And then we are trying to create new organs from scratch. Some of the ones we're looking at now are like could we make a pancreas, for diabetics and things like that.
And am I right that engineered skin grafts are already in clinical use?
Yes.
And what about the blood vessel grafts?
Well, they are in clinical use now too. One of my former fellows, Laura Nicholson, started a company called Humacyte, and I know that they're already being used in the Ukraine. And I think the approvals, and they're certainly in very advanced clinical trials in the United States.
And what do you think? What organ or tissue is the most promising to come next to the clinical use in tissue engineering?
Well, I think the blood vessel work she's doing, I mean, I think that'll get much more wide. I mean, that's at the FDA now. I think hopefully that'll get more widespread clinical use. I mean, I think there's certainly others. I'd love to see a pancreas someday, become in clinical use. So there's a lot of work going on that, not only in our lab, but other labs too.
Mm. Professor Langer, you've discussed the need for engineering to gain more respect in the United States. Have you noticed any changes in that regard?
Well, I think engineering is respected, but bioengineering, it depends. Ultimately, respect is by who. I think it really depends on the situation. So I certainly think engineering is respected, but I mean, just to take a step back, science in general, people have made this kind of comment that a society, gets what it celebrates. So I do think in the United States, what's celebrated is like actors and actresses and maybe athletes. Engineers and scientists in general aren't not celebrated like that. So obviously, I think it would be great if they were celebrated more, not only in the United States, but anywhere.
Mm-hmm.
And do you think anything is already done to foster this?
Anything?
To foster the gaining of more respect of engineering and science overall.
I think sometimes things are done. I think every so often there are awards that come out and get a certain amount of publicity, but they certainly don't get anywhere near the publicity that sports or entertainment get. But I mean that's just the world we live in. I think in terms of people's values.
And if we go back to your lab, what is the most recent breakthrough your lab has achieved?
Well, I like to think that the work that Alice Stanton has done, we're collaborating with Li Wei Sai, she's created this brain on a chip, which I'm very excited about. It's more that it will open up the door to study diseases like Alzheimer's and maybe ALS and things like that, so it's still, early and it's just different kinds of components, but I hope that something like that will, enhance people's understanding of brain disease someday.
Mm-hmm.
And if we go back to 1974, you started working with Dr. Judah Folkman on inhibiting blood vessel growth to stop cancer. In 1976, you published your scientific paper on the first angiogenesis inhibitor. It took you a very long time, 28 years to be exact, to get the first anti-cancer drug approved. So are there any ways right now to speed up the process of translating research into commercial applications?
Yeah. Well, so we published a paper on angiogenesis inhibitors, and the first one, like you said, it wasn't me ultimately that did the first one. That was actually Genentech. I mean, they built on some of our work, but I give them tremendous credit, and Napoleon Ferrara, who worked there, tremendous credit for doing that. And that was 2004, 28 years later when it got approved. In terms of speeding things up, I mean, emergencies speed things up, right? Another one that I was certainly involved in, and this is the opposite, was the COVID vaccine. I helped start Moderna, that got approved very, very quickly, but it was, people were dying all over the place. So that speeds it up. I don't know that we want that.
Mm-hmm.
And what was the reason that with COVID-19 vaccine it went really fast?
Well, I think there are two things. One was the technology, messenger RNA technology and nanoparticles. That was a much faster way to do things than, classical drug discovery. And of course, that built on various people's work, and a lot of things. So, but the second thing, of course, that speeded it up was the crisis. the regulatory authority, people put more money into it, like Operation Warp Speed, and also the regulatory authorities move faster too, just because people were dying so frequently.
Mm-hmm. And if we're already talking about Moderna, let's talk about you. Apart from academia, you're also an incredibly successful businessman, and you have founded over 40 biotechnology companies, including Moderna, a well-known American company specializing in RNA therapeutics. And from your perspective, what were the easiest and the most challenging parts of founding and growing biotech companies like Moderna?
Well, the hardest thing really is, for me, is finding good business people. We were very fortunate, like in Moderna, that Nubar Fayan, who was actually an MIT graduate, and I was on a thesis committee, started a very successful venture firm called Flagship Pioneering, and he was the chairman. We got Stefan Mansell, who was a really great business person as a CEO. Stephen Hogue, who was not only a good business person, but an MD and a really great scientist as president. Having great people, I mean, that's always probably the biggest challenge. And particularly for me, as a scientist, having great business people. Because, I mean, science I can help on. Business is, I'm not a businessman, really.
Mm-hmm. And talking about people, hiring suitable people is one of the most important parts in creating new companies, I guess, research groups and also laboratories. Some employers believe that accepting a generally nice person with a few missing necessary skills is worthwhile as long as they are motivated and hardworking. What is your opinion on that?
I'm not sure I understand everything. I mean, I think you want great people who are nice, and you want them to be hardworking. I mean, I think you want everything.
Yeah, but if they don't have a full set of skills that you're looking for.
What does that mean, full set of skills?
I think this is how we think it goes. If you're looking for a person to hire, you have a particular skills that person should be able to do.
I see.
So, and if this person lacks some of these skills, but they're important, would you still consider this person if he's like a hardworking?
Yeah. Well, I the way I always think about it is I'd rather, I use this analogy, you want a great athlete. And if that athlete isn't specialized in a particular area, to me, that's fine. I'd still rather have, somebody who is a superstar in general than a specialist in something, if that's the question.
Mm-hmm. So Professor Langer, we would also like to know, how do you stay creative throughout the hundreds of projects that you have successfully completed or not successfully completed, or in that you are currently running?
Well, I feel like a lot of it's having just great people around me, great students, great postdocs, undergraduates. I think that it's really the people again. I mean, I read a good deal, but I mean, but it's talking to all kinds of interesting people around me.
And so it comes from other people to you, and then you get ideas from them?
Well, sometimes I get ideas from other things too, from reading, from watching TV, from listening to music. I mean, ideas can come in all kinds of ways. So, yeah, so it's a combination of everything.
And as you said, you're reading, you're watching something. So you have, first you have so many projects that you're currently working on, and you also tell that you exercise daily. But I think you also should have some time for winding down. So our question is, what is rest for you personally? And do you think it's important to have some time to relax?
Yeah, I do. I feel it's important. Well, I exercise a lot, like you said. Some of it's I walk a lot. That, when I was younger, I'd run a lot. Now at 75, almost 76, I do more walking. I walk with my wife, but sometimes I walk by myself and listen to music, and I do exercise a lot. But for relaxation, I also spend time with my children, and we have places, like a place in Cape Cod that we'll go to for a part of August, and things like that. So I do, yeah. But I always work. too. I'm not good at stopping.
And in one of your interviews, you told that when you were going somewhere to receive an award or something for work, you sometimes took one of your children to spend more time with them. Are you still doing that?
Yes, I do. Actually, it's amazing. Now they're in their 30s. But yeah, I am getting an award in Norway, actually. And at the beginning of September, I think 2 of them are coming.
Oh, nice.
And my wife.
Sure.
But it's a good award, so I guess the King of Norway is coming, so they'll.
So now that we talked about some of your key breakthroughs and moments in your scientific journey, I would like to ask you if you could share a specific breakthrough or a project that you consider the most significant in your career for society.
Well, I suppose the one that you mentioned before, the taking tiny particles that could deliver large molecules, I suppose that probably would be the biggest one, I suppose, in retrospect.
And why? Of course, it was very impactful, but do you have any other personal reasons for that, maybe?
Well, first, it enabled me to solve a problem that I was working on as a postdoctoral fellow, which was actually isolating those blood vessel inhibitors, showing that could be done too. And then over the years, that I like to think that work helped grow that field of drug delivery, I mean, to the point of where it is today, and which makes a big impact on a lot of different things, whether it's messenger RNA therapeutics or almost any kind of therapeutic.
And also, if we go back to messenger RNA, could you please explain how exactly it works, for example, in vaccine?
Sure. So there's a central dogma called DNA makes RNA makes protein. So a lot of vaccines, what happens is what you do is you want to have a protein that is essentially a part of what I'll call the bad guy, the virus. And so what you want to do is, if the body is going to get that virus, you want to have it get destroyed. So if we were able to give RNA, put it in a nanoparticle, give it to a person, it will make a protein. So we're just picking COVID, for example, vaccines. The bad guy, the virus, has what's called a spike protein. So what we do is we make the RNA, so it makes the body make a little tiny bit of the spike protein, but not the virus, but makes the spike protein. And then the body in turn starts making antibodies and making immune cells that will destroy that spike protein or anything associated with it. And so if you then got COVID, which you didn't have before, well, the antibodies and the immune cells, they'll destroy it.
Mm-hmm. Mm-hmm. Professor Langer, and if we go back to regenerative medicine, could you please tell us what emerging technologies or trends in regenerative medicine do you consider the most curious right now?
Well, I think 2 things. I think different tissues that people are trying to make, whether it be a pancreas or spinal cord or almost anything. And then the other thing is recreating some of these tissues in vitro, meaning like a tissue on a chip.
Could you please explain a bit more about organ on chips? What are they?
Yeah. Well, could you mimic on a microscope slide? I mean, chip is sort of almost a phrase. But let's say, so first of all, right now, what people do when they want to test drugs is they'll test them on an animal or test them on a human. And usually that takes a long time. Most of the time it doesn't work. And that's also one of the things that makes drug discovery so slow. And not to mention that it's not good for animals or people. But if you could recreate the tissue that you're trying to treat outside the body, well, that would make it much, much easier. You could then test all kinds of drugs. You could make thousands of these little chips or thousands of these little microscope slides and test them. But you'd have to make sure that whether it was a brain or a kidney or whatever it was that you were trying to test, that that when you put it on the chip or when you put it in the microscope slide, it would behave the same way as it would on a person.
Yeah, but how is it even possible to do that? Because, for example, the brain itself, it's very complex organ, and how it is possible to include, maybe, of course, not everything, but most of the important things.
Yeah. Well, so what Alice, our student, has done is she's taken, let's say you wanted to have your brain on the chip. She could take a fibroblast and, a cell from your body, convert it into what's called a IPS cell. That's an induced pluripotent stem cell. But then there's cocktails that you can give to convert that cell into each of 6 different brain cells. So you have those brain cell types. She then found a matrix that makes them organize and sort of recreate a lot of the structures of the brain. And so that's actually what she's been doing. And then she did tests to show, well, you get similar transport of molecules, on this as you would, because there's literature on what they do in the brain. And she's looking at different disease states and, also, so that's basically how you try to do it.
And what about genetic variability? Because you told me that, for example, if she creates one organ in a chip for me, but then there are a lot of other people, then it goes more to personalized medicine, or?
Well, that's a good question. I think we'll have to see. I think, but different people have different issues. We were just over on Friday meeting with the people at Massachusetts General Hospital who are doing Lou Gehrig's disease, ALS, and so, I mean, some people have diseases like that. So if you could recreate something like that, you might come up with new ways to treat it someday. Same thing with Alzheimer's.
Mm-hmm.
And now, Professor Langer, if we go back to your job as a professor, how do you mentor students to overcome their academic and professional difficulties?
Well, I guess I try to encourage people to work hard, to take risks, and not to worry about failure. I think failure is just a natural thing. Sometimes you're going to fail. That's okay. And I try to look on the bright side of different kinds of things. What I do. When I worked with Judah Folkman, my advisor, he used what I'd call positive reinforcement. He would see a positive thing in everything and believe that almost anything was possible.
Mm.
And did you use it in your career, in your life?
Yeah, I think I do that. Yeah, I would say if you ask my students, they would say I do. Yeah.
Mm-hmm. And were there any other things that helped you to overcome all the changes that you went through?
Well, sure. Challenges you're saying that I went.
Challenges and difficulties, failures.
Yeah. Well, I think, friends, I think at different times, having good friends and family, I think that, support systems, I think, help.
Mm-hmm.
And now maybe wrapping up our conversation. Last question for you is, what would you do if not biomedical engineering?
Well, I don't know. I mean, I if it was a other scientific area, maybe I would do medicine, I suppose, which would be, but I like doing science that can help people.
Thank you very much, Professor Langer.
It's my pleasure.
We're really happy that you were able to meet us today and to give us this interview. And also, especially for me, I'm studying regenerative medicine and tissue engineering.
That's great.
And sitting in front of a person who pioneered this field, it feels incredible.
Well, that's very nice. Well, you're both at a wonderful university. It's a really terrific place. I did a virtual, honorary degree there, and it was lovely. I mean, you have some wonderful people at the university.
Thank you very much. Thank you very much.
Well, thank you. You both did a great job.
Thank you.
Very much.
Thank you.
Thank you so much for listening. We hope you enjoyed this episode of Biomed Unleashed podcast and will stay with us for the next ones.